Thank you so much for having me here today and for including me in this session. You've all been in the room for an hour. I just got to stand up to walk up here. So how about you take a stretch break for a second? I'm going to give you 30 seconds. Arms up. Do some deep breathing. Maybe a back twist and maybe took an overnight flight. I did. All right, I'll get started now. So I'm going to share with you a little bit about the eDNA Explorer. This is launching within a month. We're really excited. Maybe within a couple of weeks. We're testing everything now. So we really need your input. Okay, so thank you, Karen, for introducing eDNA. This is sort of a conglomerate of grassroots projects that use some common methods so that we can help each other get used to doing eDNA, mostly metabar coding. And it involves community scientists and lots of different managers around California, and now we have international projects as well. We developed mainly others, but I helped a little bit, develop some software called Anacapa, and we've done some Earth observation integration with eDNA to model community turnover in California, and have also different case studies on systems like vernal pools, and now have expanded our partnerships to work with all these amazing different agencies like the National Park Service, NASA, and we're thinking about how can we help managers work with eDNA data, which is why most all of us are here. So we had a project called Protecting Our River. It was focused on the Los Angeles River. Has anyone been to LA that's here? Okay, cool. Has anyone been to the LA River? Yeah, not really your hotspot destination, you know? It's not the Hollywood sign, it's not, you know, the whatever. Anyways, there's no celebrities there. Usually you find creepy stuff there. And there are all these amazing organizations that are trying to revitalize the LA River and take out the concrete and make it healthy again and restore it to something that maybe we could be creative about to make it a really great resource for everybody. And so we have brought some organizations together and did a lot of eDNA processing of over 700 samples in 3 seasons. And then the Oceankind Foundation decided that this was a really nice case study to build a web tool around making the eDNA data useful for these managers to be able to help people use eDNA in decision making. So we did initial need finding research to look at what are the barriers to eDNA adoption. And with a bunch of interviews, including some international ones, we realized that the barriers are, well, there are multiple barriers, but there's barriers upon entry, right? Like how do you set up an eDNA project? The fieldwork, not so much a barrier, that's the fun part. There's missing information that a lot of managers have where they really need to rely on a geneticist all the time. And if they want to reanalyze their data or have a question later, how do you keep that relationship up? It's a lot to maintain. And so the analysis you need to be constantly helped by a geneticist if you're a manager. Being able to understand uncertainty is difficult. And you need to be able to make decisions on what information to present, what results do you tailor for your audience. And so these are really kind of the key pressing points that we thought we could address by making web tools, where we recognize that there are 3 interconnected networks that we're really talking about. There's the network of life, right, how all this biodiversity operates. There's the network of people, and there's the network of methods. And we want to be able to help connect that for you. So we will help with the eDNA Explorer educate scientists, help plan eDNA studies, and find collaborations, help find collaborators, synthesize raw DNA data. So instead of putting your processed taxonomy tables online somewhere, you would upload your raw FASTQ files, maybe just internally for your project team to be able to look at and explore, associate that with environmental metadata automatically because that was hard for us to learn how to do, and I'm assuming it's not something that everyone wants to devote a lot of time to. But if you have metadata, we know that the more metadata you have, the more questions you can ask, and the more creative you can be. And then to compare your eDNA data with other people's data, if everyone's using different software to do the analysis, I'm thinking mainly of metabarcoding here, you're not going to be able to compare apples to apples. And then trying to present results, not just on where the samples were collected and when those collections happened, but we want to help you make beautiful infographics that you can share so that you can make some decisions on like how good your assay was, or if you want to start talking to somebody else, and share your data and results with the world. And this is what the homepage looks like. There's featured projects, the species that you can look at, and initial windows that are based on your interests, where you can maybe save some projects that you want to keep following. And you can explore based on where sample collections are happening or certain organisms that you're interested in. And your results, if you were to upload a project of your own, you'd be able to see that on 4 different pages. The learning that we're hoping people will experience here is really not so much by like a textbook of standards, but by watching what the community's doing. And we're trying to build in educational tools, maybe some stars and encouragement and guidance, so that we can help people meet those standards through community practice. And this is often how science has been done. We have peer review. This is a way of seeing what others are doing and trying to follow that and to be able to receive that encouragement and feedback. One of the things that we're doing, so I mentioned the raw data that you would upload, we'll be analyzing that with a new software coming out called Tronco, which means tree in Chamorro. The lead of that was Lenore Pipes in Rasmus Nielsen's lab. You'll see the organism list, and you can filter that on a number of mismatches, and there's phylopic images, and you can see the frequency of the total samples in the project that those organisms have been found in. So it's really attractive for visualizations. And we're trying to make well-formatted charts that show changes through time or through space, your basic alpha and beta diversity analyses, and of course comparing eDNA with GBIT, because when I find something that's endemic to China and California, right, that's always what we're asking about first when we're thinking about if we trust our results or not. So this is really of interest to us, and we've been talking with the GBIT people a lot, and it's been really great. I'm excited to talk to them here. Where are you? You're somewhere. Hi. Yep, gonna bother you later. Okay, so you might be using this to say, well, okay, qPCR versus metabarcoding, why don't they match up? Or why are they seeing different results, not maybe in the direction that I expected? How do other species observations in my county or state compare? So you can look at the project level, the state level, and the national level for the scales of overlap between GBIT and eDNA species lists. You can also validate that with other species lists, so you could filter automatically on invasive species, on rare and endangered threatened species. We have someone put edible invasive species as a list. I'm like, okay, that's cool. Yeah, let's eat the weeds. That sounds great. Okay, so taking a little bit behind the scenes, you can manage your permissions, so you could have your project be completely private, but still be able to compare your results with other people's results. We think this is useful, and also you can have what you're interested in, what your skills are, a little bit about you, so a little kind of like mini LinkedIn, but for the eDNA community and the projects that you're managing. You can help advertise events, workshops, bioblitzes that you might be running. So we'd like to also be able to highlight your eDNA resources, your standards and amazing work that you're doing. We want to be able to promote that and help educate people about that. We are not developing those ourselves. And so really focusing on how do we share all of the amazing work that's happening here. We know there's a lot of other ways to promote our science out there, and we want to help complement and integrate with them. So going a little bit behind the scenes, in case you're interested, for the metadata, we're hoping that when people use the eDNA Explorer, that they're just going to deal with minimal burden. We just need, where did you collect the sample? When did you collect the sample? What's your spatial uncertainty? What type of sample is it? And then we give you 267 variables from Google Earth Engine that, some of them are ones that you may not even knew could have any relevance to your, species richness, but you might be pleasantly surprised. And for the sequence data, we have fresh reference databases that we use that we make based on the primers that you give us. So that's using whatever in the last year was uploaded on NCBI. It's not using the reference database from 2018. And everything is easily downloaded as tables. I think this is important to have the fresh reference databases. And you can give us custom reference databases if you want. Maybe that's an area that we need to build later since people are doing so much hard work on reference databases. But we have to recognize that biodiversity sequencing is happening at warp speed right now, and it's awesome. And we also know that things are going to be eclipsed by metagenomics, and so what we're trying to build is as metagenomics ready as possible. Tronco, the software that we're using, we're really excited about because it's the only phylogeny-based taxonomic classification method that truly enables use of all NGS data. So instead of mapping your reads to a reference database leaves on a tree, you can map them to internal nodes of a tree. And so it's sort of a different way of doing classification. And we end up with, for bacteria, 10 times more classifications to somewhere in the tree, even if it's an unknown species, right? And you can also filter on mismatches and relative abundance. And we've seen massive improvements from Anna Kappa, our last software, where we couldn't see a relationship between CO1, metabolic coding, Shannon evenness, and human impact, the global human modification index. But once we use Tronco, we do see a significant relationship. And so maybe improving our taxonomy assignment will help us see new patterns where we had more type 2 error before. So we're not doing everything. We're not doing this huge list of things. We're not reinventing the wheel. We really need to work together to adopt the standards and best practices that this community upholds, while really trying to foster a good collaboration space, broaden participation in the eDNA research and eDNA information-based management world in a respectful way. And so I hope that you will consider being an early user and partner. Some of our early partners are up here on the board, but you can add your logo too. And you can go to eDNAexplorer.org and put your email address in, and we will not bombard you with any spam. So I will stop there. Thank you.